All right, the last of Mendelian hypotheses comes with the idea of what happens when we go to make offspring. And this idea ties back to Morgan's gene linkage. If everything follows traditional Mendelian genetics, those two alleles for a particular character should segregate or should separate when we go to creating gametes, so that each gamete contains one allele for every character that next generation of organisms has to have. And assuming that this is normal genetics and not linked genes, then each of those alleles should have a 50-50 shot of being exactly like the parent or being some recombination of the two parents' genetic material. And so out of this, Mendel came up with two laws, the law of segregation and the law of independent assortment. Segregation, all this really states is that you have to inherit one copy for every gene from your mother, and the second copy of that gene has to come from your father. So when we go through meiosis, those organisms, when they create their sex cells, assuming everything goes the way it's supposed to, the gametes they create should contain one of the two alleles that offspring needs in its somatic cells in the next generation. So that when we go through fertilization, egg plus sperm, that offspring now has two sets of every chromosome, meaning that they have two sets of every gene. So the combination of those two alleles from their parents gives them their phenotype. It's also going to be why when I go through and do Punnett squares, these two laws have to be in effect, otherwise we couldn't use the Punnett squares to predict the offspring in the next generation. Law of independent assortment we talked a little bit about when we talked about meiosis, and this really comes into play when we talk about metaphase 1 and lining up those homologous pairs along the metaphase plate. All your law of independent assortment really states is that when we line up those homologous chromosomes, each pair of homologous chromosomes lines up independently of the next. So I can just as easily have a maternal chromosome line up on the left for chromosome 1 and right for chromosome 1 is dad's, and the next chromosome 2, dad's lines up on the left and mom's lines up on the right. Okay, but in the same scenario, they could have all of mom's on the left and all of dad's on the right. They're completely independent of each other and of every division that happens thereafter. So if we look at this, here's sort of what we're getting at. The law of segregation simply states that I have to have alleles for each trait in each gamete, one set each. So the R and Y are in every allele. Law of independent assortment looks at this whole picture. So I could just as easily had all of mom's line up here, all of dad's line up here, so that when I get to the end, here's all mom's, here's all dad's. But it could have gone this way as well, where dad's lined up on this side, mom's on this for chromosome 1, chromosome 2 was the exact opposite, and so now I've got a mixture of that information in the next one. Alright, and when we fertilize those eggs and sperm, that's where I start getting those Mendelian ratios.